Assalamu alaikum. النهارده اليوم 4/3/8 and it is the third seminar for our summer seminar series organized by Climate Change Information Center and Renewable Energy and the US Embassy in Cairo. My name is Anjali Bamzai and I am Embassy Science Fellow posted in Cairo and also based at the CCIC, where I am enjoying the hospitality of my Egyptian colleagues at the Ministry of Agriculture. Today we have a talk by Dr. Kulvinder Gill. He is in the Department of Crop and Soil Sciences at Washington State University. So that is on the Pacific side of the US. So it's very early morning for Dr. Gill. Sabah khair, Dr. Gill. It's 4:30 a.m. in the morning for you. Thank you so much for joining us. We are grateful that you are able to share your insights and experience. So he's going to talk about how genomics, molecular biology, physiology, fast breeding approaches all come together in an interdisciplinary way. And the title of his talk is Lab to Field Translation, Technologies to Improve Heat Tolerance and Climate Resilience of Wheat and Other Crops. So this is all very relevant to Egypt, whole of our Egypt and this area, the MENA region. So before Dr. Gill starts, I will turn it over to Dr. Ala Khalil, who is the head of the Drought and Early Warning Systems Unit here at CCIC. And after that, Mr. Chuck Fee from the US Embassy in Cairo will make some remarks. And then finally, Dr. Gill will give us his talk. So over to you, Dr. Ala.
Okay, thank you a lot, Dr. Anjali, and welcome, Dr. Gill, and welcome to all of you. I am Ala Khalil, Supervisor of Early Warning Unit in Climate Change Information Center. And on behalf of Dr. Mohammed Fahim, the Chief of Climate Change Information Center, I'd like to thank US Embassy for support us and for weekly seminar. And I would like all of you during this presentation increase your knowledge and information about climate change issue and its impacts on agriculture sector. Thanks again for you, and now I give the word for Fee, Mr. Fee, and after that in the final Dr. Gill. Thanks again.
Great. Thank you, Dr. Allah, and thank you, Dr. Anjali. This is Mr. Chuck Fee from the US Embassy. And again, like Dr. Anjali, I'm pleased to introduce Dr. Kulvinder Gill. I'm lucky to have met him here in Cairo already. So he's a good friend of the embassy, in addition to being a world-renowned expert on wheat varieties. In fact, he's already working with some people inside the ARC and the Ministry of Agriculture. I'm sure he'll talk about that a little bit today, or maybe you can ask him for more information. I think we can see from the news why everyone understands why food security is so important and why Dr. Kulvinder's research is so timely to us. So we're very lucky to have him today. And, as Dr. Anjali mentioned, especially because it's so very, very early on the west coast of the US where he's based. So thank you, Dr. Gill, for joining us, and to Dr. Anjali and Dr. Ala Nassar from the embassy for arranging all this stuff in our call today. I should also mention that some of Dr. Gill's research in Egypt is being funded by the US-Egypt Joint Science and Technology Fund, which is a long-term collaboration between USAID for the US government and STDF and the Ministry of Higher Education from the government of Egypt. So we're excited that that's the case, and hopefully he can talk more about what you can do if you're interested in getting some research money from the Science and Technology Fund. And then, of course, tomorrow we have another special event. I'll be back over at CCIC to meet some of you in person, and we can talk about all this stuff about how the embassy and the US government can help you. So that's it for me. Thanks, Dr. Allah, Dr. Anjali, and Dr. Kulvinder for all the stuff you've done.
Well, Thank you so much, everyone. Real glad for this opportunity. And let me put my screen on first. Okay, so
All good.
what do can you see the presentation?
Yeah,
Yes.
we see.
All good.
Yes.
All good.
Yes.
Perfect. What I have done today is, or what I plan on talking about is about our project that we are just wrapping up that was funded by USAID with the specific target to Southeast Asia. But some of the outcomes and products are very relevant for uh, uh, Egypt. And my collaborator in Egypt, Dr. Ahmed luckily he is also on the meeting. So very nice to meet you, Ahmed, and I hope you had a wonderful vacation.
Oh, thank you,
So,
Dr. Gill.
but I will talk less about that project, but more of what we have to share that we believe is very relevant for Egypt, that we have several lines and products and technologies that can be immediately implemented or tested so that Egypt can benefit from that project as well. So that's why my talk will stay more around that project, and some of the technologies I will highlight those that are relevant. So I know this is probably a talk every day, but uh, it's just anytime I go to this Google ticker for population, it amazes me how accurate the estimates were long time. Like for example, what's shown here is, and then this number I copied yesterday from that ticker. So we are very much on track to what expectation was, that how much population will increase, where will it increase, and then unfortunately the summary of that part is that the part where development is lagging behind is the area where population is mostly increasing, and then that will always bring food security issue to the forefront. And then probably these estimates didn't really consider climate change implications into it. And when you start thinking about that, the situation, in my opinion, is very scary. So that's why I always like to talk about and remind myself and my fellow colleagues about the situation we have at hand in the coming years. And it's not like too far in the future. We are talking now that we need to address this issue. And I know some of you may not agree with what I'm going to say, that this slide is showing the grain production and then the around the globe for the three major cereal crops. As you will see that maize yields and production has been growing steadily, whereas rice and wheat for the last couple decades at least has not really increased much. And when we start looking at the yield per se, yield per acre, it's not increasing, it is stagnant. So this is really the number that if we look back in 1990, we had varieties that could yield 10 ton per hectare. Back in 1990, we had wheat varieties that could yield 9 tons per hectare. Average yield of wheat around the world is still 3 tons per hectare. And then the rice is closer to 4 tons. So while we put major emphasis on increasing yield potential, and then let's keep pushing and put more effort into breeding, which we should do. I'm not arguing against that. But what I'm arguing is that we had varieties that could yield very high long time ago, some 30 years ago. So, but we are not harvesting what the potential of that variety is. And where are we losing that? We are losing about 70% to abiotic stresses and the remaining to biotic stresses. So I always argue that we should put a lot more emphasis on protecting yield rather than increasing yield potential. So our breeding needs to be very, very targeted, address the issue that is causing the yield loss, and then hopefully that we will be able to bring major improvement into yield, because this 3 ton per hectare is not very high. For Egypt, the Egypt yields are very high, water is good, the weather is excellent. So the average yield in Egypt is quite high, but still below the yield potential. So there's quite a bit of protection that we can do, and that we should work on. And the temperature, I know that the temperature has been, or heat stress has been in the news lately, but it always has been a problem in wheat, because wheat does not like very high temperatures. Sorry, my numbers are in Fahrenheit, but what I'm showing here is anywhere there is red, that means we have, so all the analyses we have done, that we have observed significant yield reduction because of temperature during flowering, wheat flowering. Just giving you a specific example of where I'm at. So this is Pacific Northwest, around the Seattle area, and the state of Washington. We are not known for our heat stress. We believe that our temperatures are pretty low, but when we did our analysis, this is the analysis. Just want to kind of highlight that even in part like Pullman, Washington, where most of the people would never guess Pullman to be having yield stress or yield reduction due to heat stress. So what I'm showing in this slide is this green bar on the top is temperature around flowering. And the 2 bars, the red and blue below, are 2 very famous popular variety where we had a lot of data from a very reliable variety testing program that we could depend upon. And these 2 varieties are targeted for 2 different parts of the state, the red being for a low rainfall area and the blue one for high rainfall area. But if you look at their yield pattern is almost identical, and it matches almost perfectly with the temperature bar. So anytime temperature goes up, say like as we saw in 2007, 8, 9, somewhere in those years, and then the yield of these varieties dipped significantly. And anytime the temperature stays mild during flowering, as in 2010, the yields went back quite high, regardless of which variety. I mean, there are so many other factors: the soil temperature, the fertilizers, the diseases, and all those. But the yield pattern of these 2 varieties matches almost perfectly with the temperature, right? Although there is the other factors. And then another point we learned from here. So look at the start of the yield here in 1996, 97. whatever the yield was of these two varieties at the start of their production, we never recovered that yield for the next whatever number of years. So this is really puzzling us. We have done this analysis in other parts of the world, and the pattern is very similar. So over the years, the same variety, whatever its original yield was, we slowly lose that yield and almost never recover it. So then when a new variety comes, we compare with the standard variety, which has already lost quite a bit of yield. So my point being, although the new varieties show significant yield improvement over the existing variety, but if we compare that yield with when we started that variety, it almost never reached that point. So that's why, probably why, that even though we keep developing new varieties, we keep releasing new varieties, but the average yield per acre usually doesn't increase. It's kind of staying stable. So we need to look into this and then see how we can push that yield higher per acre. So talking specifically about our project, where the focus was developing heat tolerant wheat varieties for Southeast Asia. And we set very clear goal for us, that is, we must develop and release wheat varieties that could yield equal or higher than the existing varieties under normal conditions, but do not lose yield when there is stress conditions. So I'm sure you have seen the press that all the heat wave coming to Southeast Asia this year, and then causing up to 30% yield reduction on the existing varieties. And I'll talk a little bit about that. So the approach we took, we were fortunate that we had a large team, significant funding for this project. We thought that there was really no one aspect that we could focus on. So we used physiology, molecular biology, whatever technology we could lay our hand on, but we stayed within so-called natural methods. We avoided GMO methods just because it was not accepted in the countries we were targeting. So that's why we put major emphasis on exploiting natural variation already present in the nature and bring that heat tolerance into our cultivated wheat. So we made several germplasm collections. We reached out all over the world and made a list of close to 8 or 10,000 wheat lines and systematically evaluated, because you can't really evaluate that many lines effectively. So we went to their pedigree. So we spent a lot of time evaluating those collections. This was one collection, and then we went to, so our logic was that collecting wheat from areas where heat is a reality, that is around equator, that if we collect material from there, that is expected to have more heat stress tolerance. And then, so that's what we did. So we are very proud of this collection, about 2,000 lines now. Any of you who are interested in evaluating this material for your target trait, we would be happy to share with you. And whatever trait we have looked for variation in this collection, we have found amazing variation. So I think this collection is a gold mine. We would like community to use this for their target traits, and we are specifically using it for heat stress tolerance. And then the next, I know controlled condition screening probably is not as reliable, but heat is the easiest one to control in controlled condition screening. So we spent a lot of time, came up with these 5 different protocols. Like, for example, we wanted to simulate what temperature does during wheat development, say in Southeast Asia. And then we simulated that condition in our controlled condition growth chambers. Night temperature was shown to be a big factor, while the protocol 3 is that keeping temperature at normal during daytime, but increased temperature during nighttime. In fact, this part was done by one of the fellow Egyptian friend, Dr. Marwa, Sanad Marwa. And so she did part of this work when she was in my lab for a short time. And protocol 4, if you notice, very, very high temperature, 40 degrees Celsius, right, during the daytime. We wanted to find true winners. Usually 40-degree temperature doesn't reach during wheat growth, but we wanted to see if there are lines that could survive something like this. And sure enough, we found those lines. So most of the currently grown varieties, they died under this very extreme exposure, like 40 degree, but we found lines that did pretty well at even at 40 degree, that the yield reduction. So what I'm showing here is red being under heat treatment and blue under normal. So just looking at which lines showed the least reduction. So those are marked by green dots. So this is kind of how we identified these lines. And then we sent these lines for field testing in various locations in India, on an average about 5 locations throughout India, especially in the areas where we suspected heat to be a big issue. And then the controlled condition as well as the field evaluation combination of that when we did composite analysis is what helped us identify the 19 lines I talk about all the time. So these plants were grown except for germination. So at 2-leaf stage, they were moved into the chamber where they were daily exposed at 40 degrees Celsius throughout the lifespan, and look at how happy and healthy these plants look. And I will just remind you that most of the varieties that are currently grown throughout the world, either they just died or usually set only 1 or 2 seeds. So these lines truly outshined anything else because this is a really severe temperature. And but we'll skip a lot of the detail of that. Also want to highlight that soil temperature at planting is a big factor. That's what we learned. And then just part of this, what I'm showing here is we took seed from exact same envelope and germinated at different temperatures, starting from 15 degrees Celsius to 40 degrees Celsius under controlled conditions. And look at the reduction of germination as we go from lower temperature to higher temperature. So big factor, not only that, the variation that some lines showed a dramatic reduction, whereas others did quite well. Like for example, this KSU 126, it did quite well even at 30 degrees Celsius. So we are actually really seriously looking into this factor because we rarely look at the soil temperature and then see that we just always give credit to other factors. But just the germination, if we don't have a germination, there won't be much crop in the field. But even the more amazing part was that once plant was exposed at germination to high temperature, it never forgot. So this particular experiment, sorry, it's a busy slide, but I'll try to simplify. So what we did, we germinated same lines at 15 degree to 35 degrees Celsius. And after germination, we moved those plants to normal conditions altogether. So look at the difference that some lines, for example, if you look at grain yield, this third bar, look at this line KSG 063. So it remembered that if it was germinated at 15 degrees C or 35 degrees C, the yield reduction was significant. So it makes huge difference. What was the temperature in the field at planting, and it will have significant effect on grain yield, even if the weather pattern changed afterward and the weather stayed nice for the remainder of the crop season. So we are identifying lines. There are significant variation among lines. We have found lines that could germinate at much wider range, and then we would be happy to share that material with anyone interested to pursue that. So this is sort of a summary of what we did from our evaluation. So we have identified 19 wheat lines, what we call as donor lines, which we have evaluated that these contain different types of heat stress tolerance. Because heat stress tolerance, there isn't one trait. There are several different traits involved in heat stress tolerance. So we identified these 19 lines to be carrying very different type of heat stress tolerance, with the objective that we should be able to combine different type of tolerance into a single line at a later time. And these donor lines were used with our fast breeding method. So if we talk about standard breeding method, 10 to 15 years is pretty standard from making a cross to having a product in the line. Whereas with our fast breeding method, we can have line in the farmer's field from making a cross to farmer's field in 5 years. It is not only theory. 3 of our varieties are already very popular in the farmer's field, and they were developed by this method. And several other lines were developed, and they are being tested. So 40 lines developed from this fast breeding method were tested at 8 locations in India and Pakistan. And this year, unfortunately, was the best year for screening of looking at the heat stress into these lines. So I just got a call from Dr. Khan, the Vice Chancellor of Faisalabad Agriculture University. Actually, he is making a presentation to Agriculture Minister in Pakistan. And what he told me over the phone, I haven't really seen at the data yet. I will. He will be sharing that with me. He shared a picture that our 40 lines showed no shriveling, even though the heat was so severe during flowering this year that some of the farmer field, they lost 30% of the yield. And these lines showed essentially no shriveling. And we're trying to find ways to have these lines tested in Egypt, see that if they have the type of heat tolerance that we expect them to have. And then the background, their agronomic is very good. Dr. Elcott tells me that cement material does really well in Egypt. And then the background blood in these 40 lines is cement material. So we expect these lines to be doing well in Egypt also. And we are making plans and looking for funding to have these lines tested. And because these are variety candidates, they can be tested for 2 years and immediately released as varieties if they pan out to be valuable. And then these 19 lines, I invite all the scientists on the call to please consider using these lines for your basic research, for your applied research. These have some amazing features. I will share a couple of those in this my talk. Just sort of trying to go quickly through, we put major emphasis on physiology, but with the focus that we will bring that into our breeding pipeline during the project, not 10, 15, 20 years later. So we used a simple logic that we said that to have ideal heat tolerant line, we wanted a wheat plant that could maintain photosynthesis at elevated temperature. Then we wanted a wheat line that could maintain sugar transport at elevated temperature. And then we wanted a wheat line that could convert sugar into starch at elevated temperature. The genes are different, these processes are different, these physiological mechanisms are different. So that's why we sort of had these three different aspects. And then we are individually handling these and then plan to combine those into a single line at a later time. And then we consulted scientists from around the world and made a list of these 10 genes that have been shown in different crops by different scientists to be directly involved in heat stress tolerance. And then our partners are studying these enzymes in detail with the hope to make markers that breeders can use to improve heat stress tolerance. And then any of these markers, we will be very open to sharing this with all of you or whoever is interested. We are quite open with whatever we are generating from this. And I will show you two examples of two of these genes that we are studying. One is we called RCA, viscoactivase. Won't go too much into detail, but this enzyme we have found to be a bottleneck for heat stress. So if we look at the most abundant protein, ribisco, that is very, very heat tolerant. At 50 degrees C, it can function just fine. But the enzyme that keep it active, viscoactivase, is very, very heat sensitive. So we are focusing on that. And I'll skip the detail. We have spent a lot of time studying this enzyme, and then we have found the copies, done some good analysis, expression analysis. But at the end, what I'm just sort of talking about is we have found a way to make RCA very heat tolerant. So this is really the enzyme that controls ribisco. Ribisco controls photosynthesis, and if photosynthesis is not going well, the carbon will not be fixed properly, sugars will not be made. So photosynthesis can be a major bottleneck, and controlling photosynthesis under heat stress, RCA is a major bottleneck. And we have found a way to manipulate this gene to improve heat stress tolerance. We just filed the patent on this, but the terms in our agreement is pretty clear that since the project is funded by USAID, we have very generous terms for the use of any material that is being developed from this project. And another enzyme that we are looking at, this is a heat shock protein that was identified from yeast to be involved in the heat stress tolerance. And then when we looked at in wheat, just again, long story short, we have found one of its variant that actually seemed to increase in expression. And then when it does increase in expression, it actually does more damage than good. So we have learned that if we look for varieties that do not show this increase in this enzyme, usually they are more heat tolerant. So we think we have a good marker, but it's expression-based marker that we can use to screen for germplasm and breeding material for heat stress tolerance. And this work is from our collaborator, Dr. Vara Prasad and Krishna Jagdish at Kansas State University. Now Krishna moved to Texas. But again, this is a very busy slide. What I'm showing here is the vital differences for pollen viability, because again, the plant may be doing amazing photosynthesis under heat stress, but if pollen is not viable, we won't harvest much. So we have found a line, if you look at the bottom, this KSG 1186, it shows zero reduction in pollen viability at high temperature. So several of these lines, like the listed at bottom 3-4 lines, they showed very little reduction in pollen viability under heat stress. So we have identified lines that could maintain photosynthesis at elevated temperature. We have identified lines that could maintain pollen viability at high temperature. We have identified lines that could maintain starch production at high temperature. So we are in the process of combining those into a single line to further push heat stress tolerance in wheat lines. And toward the end, I will share this very exciting technology that we have developed. So if you look around when you are driving through countryside, you will see crop being affected by disease, crops being affected by heat stress, drought stress, but you will not see weeds being affected much, because those weeds who get affected much, they get eliminated. So the wild relatives of crop plants have tremendous potential for useful genes, the disease resistant genes, the drought resistant genes, heat stress resistant genes. But in wheat and other crops, the chromosome pairing control genes like pH would not allow transfer. So gene transfer is nothing new. People have been trying since 1950s, that they saw the value in wild relatives of wheat and other crops, that all the valuable traits they have, and they have been transferring. Making cross was easy, transferring complete chromosome was easy, but one chromosome of wheat is on an average close to 6,000 genes. We only need one gene. And there wasn't a good method to transfer single gene from wild relatives into cultivated. All the germplasm that people have developed, hundreds of lines, to my knowledge, only 1 or 2 are in production or in varieties. The rest of the germplasm stays as germplasm because breeders would not use those lines because of unwanted material that comes with the complete chromosome or half chromosome or even large segment that gets transferred. So this new method that I will talk to you and show you a little data about is that we can transfer chromosome segment as small as 40 kb, which is typically 1 to 2 genes in wheat, without any other material. And based on this, we have set up a new company, Biotech Naturale, and just to commercialize this technology. And we were fortunate to get several grants on this. So we have shown this to be working in wheat, and now we are showing it in soybean, in chickpea. We have reasons to believe that this technology will work in essentially all crops that we test. So, and this is based on this gene we have cloned, this pH. And although there's still some more work to be done in characterizing this, but we are focusing on using this for crop improvement. And then this whole process, the best part of this is non-GMO method. The entire process takes, let's say if there is a gene known to be present in a wild relative, we make a cross, and then the transfer, the entire process takes less than a year, because so what we do is we make the cross and transiently silence the gene using virus-induced gene silencing. And then next generation, the virus is gone, the gene function is normal, and then our transfer has happened. And some of that data is shown here. Red is rye, green is wheat. So we crossed a wheat line carrying complete rye chromosome, chromosome arm, and tried to break that arm up so that we can transfer a single segment or single gene. So these are what are marked as wigs are some of those recombinant lines. And then see that there are these very small segments, rye segments present. In the next generation, the remaining rye chromatin, we managed to segregate that out by simple segregation, and then could maintain or recover only single gene transfers. And then, so this technology we are putting massive emphasis on, and all while at WSU. So this work was originally done at WSU, and then we got this patent transferred to a company, Biotech Naturale, and now this is being done at Biotech Naturale. And then we are trying to transfer scab resistance, drought resistance, and several other valuable traits from wild relatives that are already well characterized and well known for carrying valuable traits. With that, just a quick summary that we have developed a new method of transferring genes from wild relatives into crop plants. And we could transfer single genes without any unwanted genes. Then we have shown in 2 crops, and then done analysis. And then based on that analysis, we have reasons to believe that this method will work in essentially all crops. And then the entire process can be completed in about a year's time. It is a non-GMO, thus no regulatory hurdles. And so we believe that this method is poised to revolutionize crop improvement, because, like, for example, stripe rust. When we visited wheat fields, we noticed that how badly stripe rust is damaging wheat crop in Egypt. We have wild relative genes identified, even partially transferred into wheat, but none of those are in varieties yet. The reason is what I said earlier. Those are complete chromosomes, large segments, and those unwanted traits that comes with that valuable gene will stop breeders from using that material. So with this technology, we should be able to clean up those lines and get rid of unwanted chromatin, and then just have only the gene that we are interested in. So with that, I will stop here and entertain any questions you may have. And so my email is given below. So please feel free to contact me with any questions or comments.
Hello.
Hi, Wael, please go ahead if you have any question.
Okay. I would like to explain something that's not clear for me. How it become? You say that's not GMOs, but you are transferring a single gene, and as what I think in plant as in animal as well, the gene in the gene network. There are a gene network, and there are other side effect of transferring this gene, single gene you were speaking about. So how it come to not be GMOs? So under regulation of GMOs,
right?
Thanks.
So Right. So the regulatory definition of GMO is that when you take a gene from a different organism and artificially transfer into another crop using transformation type methods, that's called GMO. So this transfer happens by crossing. We do simple crossing. So crossing is non-GMO, and we used plant's own machinery recombination to recombine and keep the gene. So basically, this is a gene replacement, not gene transfer. So a bad gene present in crop is replaced by a good gene from its sister line. That's why it is not GMO. So it is not a foreign gene coming into wheat. Its a corresponding gene is already present in cultivated, but that gene is ineffective. So we replace that gene by transferring it from one of the sister lines, and that's why it is non-GMO.
Moving to you, Dr. Ala, you have a question?
Okay, thanks, Jill, for your presentation. My question, you talked about each degree increase above 30%, 30 degree, this make a reduction for 3%.
Right.
This for your experiment or for general in global?
So this original number came from CIMMYT. And right, so in our analysis also, so as I showed that there is tremendous variation from line to line, from region to region, but we can, based on all the data and published not only from our group, but from CIMMYT and other organizations, that actually CIMMYT says that any increase over 28 degrees Celsius cause this damage. So so that is so it's quite well accepted number that damage happens. And then we also know that whatever the potential of the variety is, then often we do not recover that, and then abiotic stresses are a quite big contributor to it. And in our controlled condition, we have seen lines like when we screen the germplasm, some lines will suffer essentially no damage, others will suffer significant damage. So there is quite a bit of genetic variation in the background. But since CIMMYT material rules the wheat production in the world, and their numbers probably will apply to more wheat growing areas than not.
Thanks. Another question, please, Ala.
Sure, Dr. Ala, please go ahead.
Do you calculate the water requirement for during your experiment under different temperature? How much water?
Great question, and I'm pretty sure that Ahmed will not be very happy that I didn't talk much about our project that we are doing together with Ahmed. So our project that is STTF and USAID funded project that is in third year is targeted exactly at that. So what we wanted to do there was, because in Egypt, it is not a drought because nothing will grow without irrigation. It is what I call water use efficiency, that how much grain can you produce per unit water. So our goal, Ahmed and I, we spent a lot of time discussing and came up with that if we can cut one irrigation, that's 20% water saving, right? And Ahmed has his hand up, and if he speaks, he can give you the first-hand data that we believe that it is doable even with the currently grown varieties. And then with our water or drought tolerant material that we are working on, we may be able to further push the water reduction or water consumption on the wheat production. So there will be a direct link between temperature increase and water requirement. So evapotranspiration, those are obvious things. Do we have clear data on that? No, we don't, because we have not tested that under controlled conditions yet. We will, we are planning on it, but we do not have that data yet. And Ahmed, would you like to say something more about that cutting one irrigation, that what our data? So Ahmed has 2 years data on that. And so Ahmed, if you could please comment on that.
Okay. First of all, I would like to thank Ala for coordinating this session and US Embassy for conducting this seminar. And thank you very much, Dr. Gill. It was very surprised to me to see you today. Since I know I was in vacation for 5 days, but I'm very happy to see you and sharing the great knowledge about your heat tolerance project, which has really very good impact in India and in Asia. Yeah, for our project, indeed, as Dr. Gill mentioned, we before we transfer the new dwarfing genes to elite Egyptian wheat cultivars, Dr. Gill, after long discussion, decided with me to just evaluate the elite Egyptian wheat lines under different water regime. And we select 4 different agroclimatic zones like Nubaria, Al-Matana, Sits, and Ismailia as a new reclaimed land. And we evaluated all the climate parameters like the amount of rain, heat max, and the maximum and the minimum temperature, wind speed, and others. And really, our data shown in Matana and Sits a very great result. Like cutting out irrigation on wheat, it doesn't affect the yield. Then right now we are, I think, about to finish our first manuscript. I think by end of August it will be submitted, and we will show in this manuscript the clear data about 6 Egyptian cultivars in 4 different locations, and how cutting one irrigation of wheat will not affect the yield, but even improve the yield in some location. And I hope this year I could travel to USA, and then we'll conduct the evaluation of generated BC1F1 population in Dr. Gill's lab. And after that, absolutely, we'll brought the selected lines to Egypt to be evaluated in different agroclimatic zone. And we truly, Dr. Gill and I, thank really US Embassy and STDF for support, and absolutely Minister of Agriculture, Agriculture Research Station, Agriculture Research Center for giving all the facility in different agricultural research station. And I think Dr. Gill has visited already 3 of this station in Matana, Sits, and Nubaria, and I think next visit he will go again to Ismailia. And we will share this result. We'll try to publish it in open access journal to make this result available to all wheat breeder in Egypt. Then it will be very easy to access for this data. And we will try also to make all our data publicly available. Then any scientist in any university or institute can really get good information from our data. At the end, I'm very happy, I'm very lucky to work with Dr. Gill. He is my good friend since 2012, and I consider myself as his student because he's a very, very great scientist, worldwide known, and absolutely he has a very big name in USA and in India, and he motivated me a lot when I was PhD student. And yeah, this is how it works. And now we are working together, and I'm always looking forward to continue the success story with Dr. Gill and his group in gene shifters or in Washington State University. In both he's working, and anyone need anything from Dr. Gill, please just contact me. I'll give him you Dr. Gill contacts, and sure we promise Dr. Gill and I we will make our data available very, very soon in open access journal. Right, Dr. Gill?
Yes, and I would just want to stress one point. I'm all the bit hesitant to claim, but both years as Ahmed mentioned, that we have noticed yield increase by cutting one irrigation. We are not totally surprised because we believe it may be root diseases, it may be other factors, it may be leaching of nutrients. We do not know why, but we have at least a couple of occasions we have actually observed yield increase when we cut one irrigation based on what is currently done on farmers' field. If we cut one irrigation, we have seen yield increase, but almost at all locations, we saw no reduction in yield by cutting one irrigation. And that is on the currently grown 6 varieties. And once we bring our new dwarfing genes, I have no doubt that we will be able to push to irrigation once we introduce that. So I think that will be of tremendous help, saving a lot of water for Egypt and for the rest of the world.
Yes,
Yes.
for sure.
Thank you so much, Dr. Gill, and thank you, Ahmed. If you can share your email with us in the chat box, that will be much appreciated. Thank you so much. Moving to Joanna, she has her hands up for a while. Joanna, can you hear us?
Okay, good day. This is me, Dr. Mahmoud Sulaiman from Jersey.
Oh, okay. Sorry, the name is Joanna.
Joanna is the name of my daughter. Okay, no matter what. Actually, my question to Dr. Gill about the maize, especially the yellow maize. I worked for a long time with the yellow maize between Ukraine and Egypt. And we have an uncompleted project. We want to improve the starch as a commercial trait from the yellow maize in Ukraine. We had 10 pure strains from Ukrainian yellow maize and 2 pure strains from white maize in Egypt. We care about the starch as a commercial trait. We try to transfer, but not genetic engineering, during natural selection, the genes responsible for starch from Ukrainian maize to Egyptian maize, to improving the yield of Egyptian maize. My question is, can the soil composition and the temperature affecting the genes by transferring from pure lines to another by genetic engineering or not?
The short answer is I do not know, but I still can share a lot of information. So my good friend Craig Morris, who was director, unfortunately he passed away suddenly last year. He was director of quality lab. So US government has 4 national quality labs in the US, and then he was director of one of those 4 quality labs. So he was running this, what he called GYE study, that he would collect quality samples from 22 locations every year and do the analysis. And then the purpose was to address exactly what you're asking. Does different environmental conditions, different soils, different temperatures affect quality? So his short evaluation or summary was, yes, there would be year-to-year variation, but the variety ranking will not change. That a variety with the good quality trait will stay good over the years. The one with the not so good will stay not so good over the years. So very rarely did he see crossover, that is a variety being good, because he will evaluate starch quality, he will evaluate protein, he will evaluate all. So I think close to like 30 parameters is what he evaluated. So the summary of that is that the genetic potential overrides environmental conditions, although under different environmental conditions, there will be change in quality, but relative to other varieties, it will not change much. So the pattern would stay the same. The absolute numbers will vary from year to year. I don't know if I answered your question.
Thanks. Thanks a lot.
Yeah.
Thank you so much, Dr. Gill. Moving to you, Dr. Shireen, if you have a question.
الو everyone دكتور شيرين مع حضرتك. الو
Yes, we can hear you.
Yes, we can hear you.
You can hear me. Many thanks for you. First, second, I have a question. What's the difference between this technique and CRISPR technique and the benefit of it?
Right. So the CRISPR is actually changing the sequence of a gene that is present in a line, and the transformation is involved. The CRISPR technology is involved. Whereas this method that we have developed, it doesn't involve any transformation. It does not involve any plasmid, none of that. We have a bad gene in a popular variety. We can replace that by natural recombination, not by any so most important part of our technique is that we maintain the gene order as it is present in wheat lines. We do not change that. If we transform, then it will change that. That's one. Second is, so the sequence wise, CRISPR is also changing the gene sequence, and so is this technique. But almost all the CRISPR products, they would be disrupting a gene rather than replacing a gene. I mean, there are several papers reporting replacement of a gene with the sequence of your choice, but at commercial scale, it is still not a reality yet. There may be a product or two that I'm not aware of, but most of this is that you have one target, you change few bases there, and most of the time the amino acid sequence changes either disrupt the gene, that is more likely, or occasionally it will change one or two amino acids that, like for example, herbicide tolerance, we can select for that trait by that the ideal amino acid change, and then we can select for that type of change. Otherwise, most of the time it will create stop codon or disrupt gene function. So that's sort of the general difference between these two technologies.
Thank you so much. Thank you.
Thank you so much, Dr. Gill. It has been amazing to hear all of this new information. It was all news to me. I'm not an expert in this field, so it was all news to me, and I'm really happy to hear all of that from you. I will ask if anyone has any more questions, please raise your hand now. I can't see anything in the chat box or in the participants box. Okay, I will thank you so much for your speak today. It was amazing, and I hope to see you soon in Egypt, inshallah. And I was going to ask if you are coming for COP27 or not.
We'll try. Please send me the information. Then, I mean, we have the project on. So I think I have traveled 3 times in last couple of years, and then planning on another trip or 2. So. Please share that information, and then we'll see if we can make that work.
Okay, thank you so much again,
Yeah.
and thanks for everyone for attending this session today, and see you next week, inshallah. Thank you all. Have a nice evening.
All right.
Bye-bye.
Thank you.